Hi, um, thanks for your interest in our research. Let's begin. And let's begin by the end. So, what we found is that, surprisingly, autotrophs are present in the high turbidity waters of the mouth, where we were expecting that light availability will limit primary producers. Then, Mixotrophs appears related to dinoflagellates or what it seems to be diatoms fueling a complex food web with a strong microbial loop and protist heterotrophs. And the Amazon is one of the largest rivers, and it produces, or its plume extends thousands of kilometers to the north, and it has a seasonal component. So it has a peak season that begins in boreal spring and extends until late summer, early fall. And as you can see here, those bluish colors that increase and basically uh, spread to the north. And so it is very likely that while the plume travels to the north, it defines different habitats with different trophic structures. And trophic structures are essential for defining how much energy reaches upper trophic levels, so top predators. Why? Because the higher the trophic steps, as you can see here, the lower energy reaching those top predators, because only about 10% of energy on average goes from one trophic level to the next. So it's important to define those because in the marine environment, many times top predators mean fisheries. So we've been in the area of influence of the Amazon River plume in 2021 at the beginning of the peak season, and we sample all these stations that you can see here. And as you see, they are colored by habitats, and in a fashion, they are here ordered by age of the plume. So we begin in the mouth where you see all these gray dots that belong to the riverine input, where salinity is low, turbidity is high, and then there's the next step of aging of the plume in the jump plume core, and this follows until the modified oceanic water, which would be the last stage of waters or age of the plume or waters affected by the plume. And then we have the oceanic water in dark blue that are waters with no influence of the plume at all. And there's a manuscript that is going to submit soon explaining how this approach was done. But basically, what we did was using city-derived variables, and you see them here in the upper left corner: sea surface salinity, sea surface temperature, mixed layer depth, the depth of the deep chlorophyll maximum, and a nitrate availability index. We apply a hierarchical cluster analysis to them and found 8 habitats. But for this study, we only got samples in 6 of them. Then we also took samples for the analysis of pigments by high performance liquid chromatography. We calculated the pigment to total chlorophyll ratio and apply a chemotaxonomy analysis following the Chemtax recommendations. And we defined these seven major groups that you see there. In selected stations, we also ran samples taken from the clean water supply of the ship into a net for the 10 to 200 microns plankton community through a plantoscope for taking images. And then the core measurements that we do are the amino acids in suspended particles. Basically, we measure the concentrations and the nitrogen isotopes in these amino acids that you see in the table, ordered as trophic and source. And for this work, we're going to use alanine and glutamic acid as trophic and phenylalanine as source for calculating different indices and proxies. For instance, the trophic position according to Chikaraishi and colleagues using glutamic acid and phenylalanine, and then the apparent nitrogen isotopic fractionation of phenylalanine versus glutamic acid, and a degradation index. So we separated our cestone, our suspended particles, into two size fractions using 3 microns as the threshold for having the phytoplankton and free living microbial community in the small size fraction, and then a mixture of nanoplankton, microplankton, and particle attached microbes in the large size fraction. And what is this trophic versus source amino acids? So, well, the trophic amino acids are those that whose nitrogen isotopic signature changes drastically when they go from one trophic level to the next. And the source are those that barely change, so they are always reflecting the signature of the nitrogen source that primary producers use at the base of the food web. And when you plot them like this, so here we have phenylalanine in the x-axis and glutamic acid in the y-axis, we can define these dashed lines, which are trophic lines, and they represent the same trophic level. So here we have TP1 for primary producers, TP2 for herbivores, TP3 for carnivores, and we keep going up the food web. And intermediate states as mixotrophy and omnivory will be like intermediate levels in TP1.5 and TP2.5. And we can calculate that using the nitrogen isotopic composition of these two amino acids. And then, so take a look at the data. So here we have the degradation index that we calculated with the mole percent of the amino acids versus the bulk carbon in the particles. And they are separated by size fraction, and you will see always a triangle for the large and a circle for the small, and color by habitat. So remember that we go from the gray freshest plume to the dark blue no plume influence. And what we can see here is really striking is that all the gray points in the riverine input habitat in the mouth, they have a clear signature of carbon related to terrestrial material. They are relatively more depleted than the rest. And then there's a quick mixing, and in the first stage of the aging plume, which is the young plume core in red, we see basically marine signatures. And the other thing that we see here is that most of this terrestrial material in the mouth is more degraded than that we find in the other habitats, because this degradation index is really negative. According to in terms of the TP of our samples, so here we have in the dashed lines represents the trophocline of autotrophs and herbivores, and in the dotted lines we have the limit, the area where we expect mixotrophs. And as you can see here, and as we say surprisingly in the mouth, we have clear autotrophy. There are some deviations because they don't fall exactly on the line, but the dominant function of these particles is autotrophic TP1. And then we only have 5 samples in 3 different stations that were clearly mixotrophic. So the large size fraction in the outermost station in the riverine input, plus 2 other stations in other habitats. And so let's try to explain this. First, we can take a look at the composition of the phytoplankton community using the pigments, and we see that the groups are relatively similar in the 3 stations, but in the riverine in station 8, so the community is dominated by cryptophytes in yellow, and in the other 2 there's a clear dominance by diatoms in green. But the other feature that they share is that there's a higher proportion of dinoflagellates in the 3 stations. So we suggest that these dinoflagellates in station 8 will partially explain the mixotrophy that we find in the large size fraction. But then it's striking, or it's intriguing, that we have clear mixotrophy in both size fractions in the other stations when diatoms are dominating, and they are supposed to be strict phototrophs. So the plantoscope images confirm the classification that we made with the pigments, and you can see, unfortunately, we don't have images from station 8, but you can see that in 12 and 10, there's a mixed community of diatoms and dinoflagellates. And luckily, we have a video from the preview of the plantoscope while it was measuring the samples at the station 12. And as I say, you can see there's a really rich, very varied community of diatoms and lots of dinoflagellates that are passing by. So, and in some cases, you can see them swimming. So this is what we have in terms of the composition. So why the next thing that we're going to do is comparing 2 proxies derived with nitrogen isotopes of the amino acids. So we saw this diagram before. This is the original one. Here, these authors try to stress the different behavior of two trophic amino acids when they go up the food web. On the one side, glutamic acid, which is the one that we are using for calculating the TP, and then alanine. So in a classical food chain, like the one that you have on the left, all phytoplankton are considered primary producers. So both alanine and glutamic acid will give you a TP1, and then they are grazed by microzooplankton. But then we know that especially protists are more complex than that. So there's the possibility that some protists are heterotrophs, and then therefore they are basically occupying the space of grazers. So the first step of herbivores. And in terms of glutamic acid, the signature doesn't change, but alanine is enriched when protists are acting as grazers. So when you have heterotrophic protists, the TP that you calculate using alanine is going to be 2, while glutamic acid is going to be 1. But we know that in the marine environment, there's no pure feeding mode, and most protist heterotrophs are in reality mixotrophs. So what we have decided to use is the difference in between alanine and phenylalanine as a proxy for this protist heterotrophy. And then the other thing that we're going to use is the apparent nitrogen isotopic fractionation of phenylalanine versus glutamic acid. And this is just extracted from the paper just as a summary. So when you have microbes that are producing their own amino acids because they're growing on inorganic medium, so their TP is 1. And this epsilon value is above -5 per mil, as you can see here in this green circle and the color dots. When you have microbes that are growing on amino acids, so they are salvaging, living on ambient amino acids, they have a TP of 2, and this epsilon decreases below -10. So this proxy can serve to trace microbes growing on organics, so microbes salvaging amino acids. So we have plotted the two of them. Epsilon is in the y-axis. The difference between alanine and phenylalanine is in the x-axis. And you can see that we have labeled our mixotrophs. And you can see that all of them show this imprint of protist heterotrophy because the difference of alanine minus phenylalanine is above the level that autotrophs usually have, which is in between 3.2 3.4 per mil. It's more clear, this imprint, in the large size fraction where we have potential mixotrophs. And then we also see that in all of them, this epsilon is below -6, so it's suggesting that there's also a microbial loop reworking amino acids. So there might be a combination of both. So maybe in the small size fraction, PQ eukaryotes are grazing a strong microbial loop, which in these two stations 10 and 12 might be thriving on metabolites produced by diatoms, because this kind of mutualistic relationships are described in the literature. So the last thing we did was trying to explain the TP using a combination of biological and environmental variables, and we used this multivariable factorial and polynomial analysis, and it selected these 5 variables. So basically, the composition of our strict phototrophs, diatoms, and cyanobacteria, and then our mixotrophs, dinoflagellated. This proxy for protist heterotrophy, and then the nitrate to phosphate ratio at surface, which in some fashion can trigger this kind of feeding modes. So back to the end. Hope that we will be able to convince you that this is what we found. And if you're interested in knowing more of what we do in upper trophic levels, there's a talk on the 7th at 4 p.m. by Steinkopf. And then there are several talks about nitrogen cycling along the Amazon River plume in session 38 on the 8th. And if you want to know what else we do in the group, just follow the link. Thanks.